I'm Teodor Aastrup, CEO and co-founder of Attana. Today I'm going to give a short introduction to Attana, followed by presenting our two business areas, research and development of new drugs and diagnostics. And finally, I'm going to explain why we are participating at this conference, what we are looking for. So, Attana, we have been around for almost 20 years. We are listed on the NGM Nordic SME in Stockholm, having a market cap around $60 million US. And what we do is that we have the third generation biosensors, and with third generation we mean that we try to mimic in vivo conditions, and thereby our idea is to improve the success rate of clinical trials in drug development. And we have an enormously good track record. We have 100% success rate of the molecule candidates that we have validated, which have entered clinical trials. So far, all of them have succeeded in their clinical trials. Our typical customers are biotech companies, research institutes, or academic institutions. So, our business areas. Our core business is to help companies who develop pharmaceuticals. So we do that in two cases. We have contract research, which means that the customer sends samples to us and we perform the analysis of it. Or you can also buy our analytical instruments, our third generation biosensors, and perform the work yourself. The other business area is a newly developed one. It's for diagnostics. It's actually an old idea of having a generic platform to meet needs of pandemic outbreaks, which have, due to the COVID-19 situation, gained a lot of interest in last year. So, our core business. The main idea behind Attana is that it's only 1 out of 10 molecules that start clinical trials that reach the market. And when we founded Attana, we were considering, why is that? Are one measuring everything? Are one measuring things correctly? Or is it something that's not measured or could be measured in a different way? And could we do that? So what we realized that there is actually no one who looks at molecular interactions at the in vivo conditions. You either do it in a very artificial environment or not at all. So our idea was to develop a biosensor where you could mimic the in vivo situation and thereby look on both target interactions, the ones you want, and off-target interactions, the ones you don't want. And that is what have made us so successful. So if you look on the drug development from another point of view, one start with several thousand of drug molecules, and then as the development proceeds, it becomes fewer and fewer molecules. And basically you need 10 molecules to start clinical trials to have one to reach the market. What we are positioning is that we validate molecules prior to clinical trials. So instead of needing 10 molecules to have one on the market, it's enough with 5. That's what we believe, that's our addition. So we look on off-target interactions to remove them and thereby increase the success rate in phase 1 studies. And we look on the target interaction to increase the efficacy of the drug and thereby increase the success rate in phase 2 studies. So how well have we been doing? Well, in December we published a study of the 77 molecules that we have performed contract research for during the last years. And out of them we could validate 30, and we could say that 47 are not will not make it. Out of these 47 that we have disqualified, to our knowledge, no one has been successful of those. And out of the 30 that we have validated, 8 has entered clinical trials, and 100% of them have so far been successful. So if you look at that into a little bit more detail, we can then see that we have 8 molecules that we have been involved in that have initiated clinical trials. 2 of them are in ongoing phase 1 studies, and 6 have all passed successfully phase 1 studies. Of these 6, 3 have initiated phase 2 studies, and 2 are still ongoing, looking promising, and 1, a molecule from Index Pharmaceuticals, have successfully concluded phase 2 and are now starting phase 3 studies. So far, all 8 that has initiated has so far been successful. So if we go back to the original to look here, so we can see that we have increased the success rate in phase 1 studies from around 60% to 100%, and in phase 2 studies from 30% to 100%. If we then would extrapolate using the same success rates in the preceding stages, we would see that we will have not 1 out of 10, actually 1 out of 2. So we have a 4-fold increase in the success rate. We are aware of that this is statistically not so many cases, and it's probably not only because of what we have done, because it's also that our customers has probably been innovative in other respects. But we are 100% confident that one of the reasons why this has been successful is because our unique ability to measure interactions exactly as they occur in the human body. So, how do we do this? Well, we have a technology covered by several patents. And first of all, our focus is on biopharmaceutical molecules. And as I mentioned, we try to simulate in vivo conditions. So first of all, the technology is of course label-free. And then we have seven different assays, cell-based, tissue-based, sera-based, blood-based, and so on, to look on the biological conditions. And from that, we generate 10 different parameters, such as interaction data as affinity and interaction kinetics, different thermodynamic data, yes or no, is there any off-target interactions, and so on. We can also have competition assays to validate and check if 2 molecules can increase or decrease the effect of a given drug. And also we can look on this interaction as it occurs in real time. And I will come back to that because that has also been one of the reasons and one of the advantages we have in our diagnostic portfolio. So what we do is that first of all we look on the molecules before we set up the experiments, because one important thing for success here is that we really study the molecules as they're supposed to be in the body. So we don't have detergents and other stuff that affects the interactions. We try to have the real clean molecules. And then we start up the instrument and we select the different assays, and depending on if you're looking for off-target interactions, which we can look in blood or on cells, on tissues, to see if the drug molecule interacts with things they're not supposed to interact with. These off-target interactions are usually weak and or fast, so they are very difficult to detect with other technologies, and if you have detergents in your assay, you will not detect them. But an off-target interaction means an increased risk for toxicological or immune response that are unwanted and hence can result in side effects in the clinical trials. So that's something we do. We also look on the target interactions, for instance on cells and on tissues, and there we can help select the right molecules and make sure that you have the optimal interaction profile for that. Then we monitor the interactions as they occur, and thereby we can get the information not only of affinity but also on the kinetics. And it's, for instance, important to know how long a molecule stay on a given receptor. And from that we basically have three outputs. One is, this is not going to work, forget it. Or, this is promising, but you need to optimize it. You might need to decrease the off-target interactions, or you need to increase the targeted interactions. Or the third case is, of course, that it's validated and let's proceed to clinical trials. So, for this we then have two solutions. Either we do contract research, and that means that our customer sends the samples to us, and in our lab our skilled application specialist performs the assays and deliver the results using our instruments and our chips. Or the customer can also buy the instrument and buy the consumables from us, and of course we provide technical support. As a matter of fact, many of our customers use both. They might have started as a contract research customer, realize the enormous benefits of our technology, buy an instrument, but still use our contract research when they need to do more advanced studies, or if they are in urgent need, or they basically need the extra hands. It can also be the reverse, that you have bought an instrument and you realize the need of doing more advanced technologies. But we offer both platforms in order to meet the requirements of the customer. Our second business area is under development, and it's actually a 10-year-old idea. 10 years ago, we realized that when we help our customers developing new drugs, a lot of that is about avoiding immune response. So basically what we are looking for is a diagnostic tool to see if you have an immune response. And we were thinking that this could be a good generic platform for diagnostics, because basically if you have any pathogen, a virus or bacteria, you can place that virus or bacteria or part of it, like a spike protein, on our sensor chip, and then immediately study the interaction between blood and the part of the virus, or in this case spike, and you can get a profile of the immunity immediately without any development. However, 10 years ago, we didn't felt that there was a market for this kind of generic platforms. But we still had made the research, and we could see that the number of outbreaks are increasing constantly in the world, and two of the major factors for that are that the population on the Earth is growing, and we are traveling more, so we have more interaction between different people, and hence it's easier for a pandemic outbreak. Now, last year with the COVID-19 outbreak, we have realized that there is a market for our technology. So what we have is that the main goal is to have this generic platform to be able to look on any virus or bacteria or other pathogen. And what is also very important is that it's enough to take one stick in the finger and have one blood droplet and analyze that. And from that we can get an enormous amount of parameters that provides information about the given person's immunity. We have validated this technically by comparing with the standard assays for COVID-19 tests. And we are now seeking a partner to develop this to a full diagnostic platform. So what we see for us is that today you basically get an answer if you're immune or not, and you can do this. You can do this kind of analysis two ways. You can test on the blood, or you can test in the nose, in the blood, to look for antibodies and other immune stuff, or you can use to check for virus. So you can see if you have antibodies or if you have virus, and from that you can basically classify a given person, whether it's uninfected, infected, or post-infection. we believe it's more to it. So what we can do is that we can take blood samples, we can take the nose samples, but we can also take biopsy and look on cells. And that means that we can not only detect, we can also look on the concentration of an antibody, the titer, and the population and the quality, basically how good it interacts with, for instance, spike. We also can look at how well the virus interact with a given cell or tissue, and thereby you can also get information on how infectious it is. So it's not only about if it is there, it's how infectious is it for a given person, and we have seen that that varies between different individuals. And from that we believe that we can then do an improved patient classification, so we can look at the person and see, are you uninfected or not, but then we can also have to predict whether you have an efficient immune system for this particular pathogen or not. So if you're not, then you are in the risk group. We also believe that during infection we can more in detail see where in the infection are you, if you are pre-peak or post-peak, and that's important for what kind of treatment and if you should be isolated or not. And then we can also see after infection or after you've been vaccinated if you have a efficient immunity or not, which of course also is very important. So that is basically our idea. And as it looks so far, we get this kind of diagram where we, first of all, and this is the good thing with the real-time stuff, is that we can measure the interaction between blood and, for instance, in this case where we compare a person's interaction with spike and tetanus, and we have chosen spike for the obvious reason that it's a COVID-19, and tetanus because, at least in Sweden, but in many other countries, the majority of people are vaccinated, and hence you can get a profile for a fully vaccinated person. So on top here you can see the non-specific immune response, and that is what you get when the blood interacts with the spike. So as the interaction occurs, you will both have non-specific and specific interactions, both strong and weak interactions. As we then let a buffer flow over, the weak interactions will vanish rapidly and the stronger will remain, and we can then measure the total amount of strong interaction, more specific interaction. We can then monitor how fast it dissociates and thereby get an information on the quality of it, and then we can add anti-IgA, anti-IgM and anti-IgG antibodies and see how much of the specific antibodies is for the given A, M and G antibodies, and thereby also look on how is the infection developing. So in this case you can see on the orange slide that this person has an ongoing infection. It actually has some IgM, some IgA and some IgG, and you can compare this person's interaction profile with the profile of that person's tetanus where it's completely vaccinated. And we believe that it's easier to set a diagnosis if you have a reference that is individual rather than comparing with different people. So this is something that we want to develop, and we also are looking for partners to do more advanced analysis of all the information we provide. At the end of the day, this is basically what we are looking for. A generic platform where you can look on different kind of infections like COVID-2, hepatitis, yellow fever, and so on, and you can get a classification of how good your immune system is, or where you are in the if you are infected or not. So you can sort of set a treatment plan, or are you well vaccinated, and it might be so that in the future that you basically need to have a vaccine passport in order to be able to travel. This is where our technology will fulfill an important role. So what we see here is that there's a market for both individuals, because basically as an individual you don't want to know if you have antibodies or not, you want to know, am I immune or not? and am I in a risk group or not, because that can determine how you should behave as an individual. It's also important that if you can do this on an individual, if you can get an answer, as we can do, provide within 15 minutes, instead of waiting 2 or 3 days as it's today, then you can also increase the contact tracing, because then if you get it immediately, it's fewer people you have been in contact with, so it's easier to trace who can be the person who infected me. For companies, this is of course important. If you have someone coming home from a travel, you can test them rapidly and see have they been infected or not, and thereby also determine whether you should be at work or not. It will also be easier to plan the working stuff. And of course then for the public sector, like now when we are trying to vaccinate people, it's important to set up the efficient vaccine strategy. It can also be good for improved knowledge on how to use in hospital treatment and so on. So where we are today is that we have a proof of concept that this works for SARS-CoV-2 and for tetanus. We in December registered our instrument as a class 1 device as FDA, and we are basically by this presentation starting to seek for partners to increase the speed in our development. What we are going to do this year is that we're going to seek FDA and EMA approval for specific assays like SARS-CoV-2 assays. We are going to continue with looking at different kind of T cells response, and we are also going to look what else would be good to have assays approved by the FDA to have this generic platform. That is our long-term goal, and for that we need partners. We also think that this will be a potential platform for companion diagnostics, and that's basically by combining our knowledge from this diagnostics and our drug development program. Because for instance, if you take cancer, we can take tissues from a given patient and test the different drugs available on the market to see which one seems to be the most efficient for this given patient, and thereby you can set in the correct treatment immediately instead of having doing trial and error. That is the case of today. And we also foresee a point of care version of the instrument in the future. So, what are we looking for? First of all, in the diagnostic platform, we look on research lab that are interested in analyzing this kind of immune profile and working with immunities and working with vaccines and so forth. We are also looking for distribution channels for this. But in particular, we are also looking for companies who can help us with analytics, because we generate so much more information that I've shown here, and we believe that with modern AI technologies one can gain much more information and have much more precise predictions of a person's immune profile. So we are really eager to start working with people who can help us analyze the data even better. And we are also looking for technical expertise in this area of diagnostics, so we can really set the platform to be a user-friendly technology out for people to use. For our core business, R&D of drug candidates, we are of course looking for people in particular working with biopharmaceuticals and who are in a stage that they would like to validate their candidates and maybe optimize them prior to clinical trials in order to increase the success rate of the clinical, or the likelihood of success in the clinical trials. We are also interested in those who have difficulties in phase 2 and phase 1. You might need to take a step back, but for instance, if we can detect a week of target interaction that potentially can be a cause of a side effect, it's definitely worth using our technology to detect it and then do some minor modifications and get rid of it and have a successful clinical trials. So with that said, if you're interested in contract research, you should contact Thomas Ljungberg, my colleague. And if you're interested in working with diagnostics with us, please contact me. So thank you very much for your attention.